host cells, a form of in vivo genetic engineering. In addition, modified mu phages have been made artificially in which some of the lytic functions of mu have been deleted. The phage called mini-mu are deleted for significant portions of mu but have the ends of the phage in normal orientation. Many mu phages are usually defective, unable to form phages, and therefore presence must be ascertained by the presence of other genes they carry. One set of mini-mu phages containing the beta-lactosidase gene of the host called the mu-lac-D4 defective can be detected in the integrated state if the lac gene is orientated properly in relation to a host motor. Under these conditions, the host cell forms the enzyme beta-lactosidase, which can be detected in colonies by using a special color indicator. Beta-lactosidase positive colonies from a beta-lactosidase negative host are thus an indication that mud-lac infection has occurred. Integrons. Because many transposons contain genes conferring both genetic resistance and the ability to transpose to conjugated plasmids, traditional antibiotic therapies and the treatment of bacteria infections can be seriously challenged. Some transposons also contain other elements that make them even more formidable. Integrons are genetic elements that can capture and express genes from other sources. Integrons contain a gene that encodes an integrase. The integrase is an enzyme that catalyzes another type of site-specific recombination. Recall that the lambda genome becomes integrated into the Escherichia coli genome at the specific site by the action of an integrase encoded by lambda. The integron also contains a specific DNA sequence, whereas the integrase can function to integrate gene cassettes, which are located near similar sequences on other elements, and a promoter that can then express the newly integrated gene cassette. Integrons have been found in a number of bacteria, for example, Escherichia coli, Pseudomonas, Acantobacter, Citrobacter. Often in clinical isolates, they can occur in transposons, for example, TN7 on plasmid or on the bacteria chromosome. Some isolates have as many as 5 different gene cassettes. Over 40 different antibiotic-resistant genes have been identified on such cassettes, as have some genes associated with virulence. Shows the structure of 2 integrons from Pseudomonas aeruginosa. The clinical significance of integrons and gene cassettes they capture seems obvious. What is much less obvious is the origin of the gene cassettes themselves. These are not simply random genes that can be captured, but genes bounded by specific DNA sequences that are recognized by the integrase, and genes that are apparently not expressed until they become part of the integron and can be transcribed from the promoter on the integron. Restriction enzymes. We discussed the fact that the prokaryotes have restriction and modification system, the function of which seems to be protect the cells from foreign DNA such as viral DNA, although such systems are very widespread among prokaryotes. Both bacteria and archaea, they are very rarely in eukaryotes. Indeed, the only known eukaryotic restriction and modification systems are found in viruses that in fact contain unicellular plants. Some prokaryotic viruses also have their own restriction and modification systems. Recall that the function of the restriction enzymes, restriction endonucleases, which are part of these systems is to recognize certain bacterial sequences of DNA and cut the DNA, one large class of the enzymes, called type 2 restriction enzymes. Not only recognize specific sequences, but also make double-stranded breaks within these sequences. Such enzymes have proven enormously valuable in many of the techniques of the in vitro DNA manipulation. Indeed, the discovery of these enzymes was the beginning of what came to be called genetic engineering. Many of these sequences recognized by type 2 restriction enzymes exhibit twofold symmetry around a given point. Shows that the sequence that is recognized and cleaved by one restriction endonuclease from Acinetobacter coli called ECORI. The cleavage sites are indicated by arrows and the axis of symmetry by a dashed line. Note that the two strands have the same sequences if one is read from the left and the other is read from the right, or in terms of polynucleotide strands if both are read 5 to 3 or both read 3 to 5. Such a structure is called a palindrome. A palindrome is a sequence of characters that reads the same when read from either right or left. For instance, sex at noon taxes. The term palindrome is derived from the Greek meaning to run back again. Many restriction enzymes are composed of two identical polypeptide subunits, and each subunit recognizes and cuts the sequences on one of the strands. Since the sequences recognized by restriction enzymes are relatively short and frequently palindromic, such enzymes always make double-stranded breaks. Interestingly, restriction enzymes that recognize non-palindromic sequences cut near the sequence, but not within it. The recognition sequence and cutting sites for a few restriction enzymes are given. While over 2,000 restriction enzymes with over 200 different specificities are now known, others are being sought because these enzymes are of great importance in genetic research. Consider that the enzyme ECORI can cut any double-stranded DNA that has its recognition sequence and cuts only at that sequence. This enables scientists to cut large DNA molecules into smaller fragments. Such fragments with defined termini, created as a result of this action, of specific restriction enzymes have many uses. Restriction enzymes are such important tools in modern molecular genetic research that they have become widely available commercially. A number of companies purify and market restriction enzymes with a variety of specificities. Modification protection from restriction. An integral part of the cell's restriction modification system is modifying enzyme, which chemically modifies a specific sequence on its own DNA, so these sequences are not attacked by the cell's own restriction enzymes. Such modification generally involves methylation of a specific base within the recognition sequence, so the restriction nucleus can no longer act. Thus, for each restriction enzyme, there must also be modification enzyme. The two enzymes being closely associated, for example, sequence recognition by ECOR1 restriction enzyme, can be modified by methylation of the two most interior adenines. The enzyme that performs this modification is called ECOR1 methylase. If even a single strand is modified, the sequence is no longer a substrate for the restriction enzyme. Restriction enzyme analysis of DNA. As noted, a DNA molecule will be cut at a specific sequence by a given restriction enzyme because the base sequence recognized by many restriction enzymes are 4 to 6 nucleotides long. There will generally be only a limited number of such sequences in a piece of DNA. After cleaving the DNA, the fragments generally can be separated from each other by gel electrophoresis. Electrophoresis is the procedure by which charged molecules migrate in an electrical field, the rate of migration being determined by the charge on the molecule and by its size. In gel electrophoresis, the molecules are separated in gel, usually composed of agarose or polyacrylamide. The gel is a complex network of fibrils and a pore size of a gel, which controls separation can be set by the experimenter. Nucleic acid. Nucleic acid, which is negatively charged, will migrate through the gel once an electrical current is applied, at rates dependent on the shape and size of the molecule. Small or compact molecules migrate more rapidly than large molecules. After a defined period of migration time, the gel can be stained with the compound that binds the DNA. Epithelium bromide is used as a stain. The DNA bands will fluoresce under an ultraviolet light. A typical agarose gel is shown. As a standard digestion has been applied and the size of each band is known, the other lanes contain a purified source of DNA, in this case a plasmid, cut by one or more restriction enzymes. Because a given restriction enzyme always cuts at the same site, the banding pattern is reproducible and using a standard. The size of the fragment can be determined. Such a technique can be used to generate a physical map of DNA where the sites being mapped are not genes or mutations, but the location of the restriction enzyme recognition sites. Creating physical maps in this way involves doing several digestions with several restriction enzymes, singly and in combination, and then determining how the pieces should be ordered. Much like solving a puzzle, each lane in the gel shown contain millions of identical fragments of DNA, coupling restriction enzymes, digestion of a single molecule of DNA. With microscopy allows a restriction map to be made directly from observing the cuts in a single molecule. This technique, called optical mapping, is shown. DNA fragments can also be purified from gels, which can be useful in some cloning experiments. However, there are other uses for these gels. Them involves nucleic acid hybridization. Recall from section 7.2 that when DNA is denatured, made single-stranded, it can then be used to form hybrid molecules with other single-stranded DNA or RNA, molecules having complementary or almost complementary sequences. These later molecules are called nucleic acid probes, or more simply, probes. Hybridization can be very useful for finding similar